So as I mentioned, thank you very much for organizing this wonderful symposium. We get to interact with all the other scientists. This is a great place for connecting and making new networks. And also thank you to everyone who is attending the seminar today. So today I will be talking about my recent work, which was published in Immunity, where we've shown that the endogenous glucocorticoid signaling can regulate effector differentiation and also promote T cell dysfunction in the tumor microenvironment. So as this audience may already know, in any chronic disease setting, such as chronic viral infection or in cancer, effector T cells, they gradually, progressively acquire a state which is commonly referred to as T cell exhaustion or dysfunctional T cell state. Now, these exhausted or dysfunctional T cells are characterized by various deficits in their effector functions, including loss of cytotoxicity, loss in the production of pro-inflammatory cytokines such as IL-2, TNF alpha, interferon gamma. But on the other hand, although they're losing the ability to produce these pro-inflammatory cytokines, they have an increased ability to produce the immune suppressive cytokine IL-10. And another important feature of these dysfunctional or exhausted CD8 T cells is that they express a whole lot of different types of checkpoint receptors like TIM3, PD1, LAG3, and TIGIT. So it's important to bear in mind that these dysfunctional T cells, they not only are poor mediators of tumor clearance, but by producing anti-inflammatory substances like IL-10, they also serve as a liability to an effective anti-tumor immunity. So our lab has been very interested in trying to understand the signals which are present in the tumor microenvironment and that push the T cells towards developing this dysfunctional T cell state. Now, I'm going to start off with a very foundational observation from our lab, where we showed that by just using two checkpoint receptors, TIM3 and PD1, you can basically partition the CD8 T cells infiltrating the tumor tissue into three compartments. The first one, which does not express TIM3 and PD1, is shown here. And the second one, which expresses PD1 but not TIM3. And the third one, which expresses both TIM3 and PD1. We looked into the effector functions and we found that as the cells acquire more of these checkpoint receptors, they gradually lose their effector capacity, but they acquire more of a dysfunctional T cell state. Or in other words, just using TIM3 and PD1 as a marker, you can distinguish the CD8 T cell effectors, which have different effector functions. We wanted to understand the gene programs that underlie different effector functions in these CD8 T cell subsets. So we implanted tumors into mice. We isolated the CD8 T cells. We sorted the three different subtypes of CD8 T cells, depending on the expression of TIM3 and PD1. And we did an RNA sequencing experiment where we looked at the gene profiles. Now, we found several differentially expressed genes, but the cluster that we were most interested in was cluster 1. This is because these genes had a certain pattern where they were low expressed in the PD1 TIM3 double negative cells, and they progressively increased in the TIM negative PD1 single positive cells and had maximum expression in the TIM PD1 double positive cells. And when we looked at this gene list, the gene that we were most interested in was NR3C1. So NR3C1 is a gene that encodes the receptor for glucocorticoids, and these glucocorticoid receptors are members of nuclear hormone receptor family. Now, in an inactive state or in the absence of the ligand, the glucocorticoid receptor is present in the cytoplasm in an inactive state where it is bound to inactivating partners like heat shock proteins. But once the ligand is available and it binds to the receptor, there's a conformational change in the receptor and it exposes the nuclear localization signal. Now, following which the receptor can migrate or translocate to the nucleus. And once it is inside the nucleus, it can regulate the expression of the target genes by two mechanisms. One, it can directly bind to the promoter of the target gene, and this is called transactivation. And the other one is where it can bind to other transcription factors, such as NF-kappa B or AP1, and prevent the binding of those transcription factors to the promoter of their respective target genes. So these glucocorticoids are steroid hormones, and they're one of the most potent immune suppressive agents known to man. And they've been widely used in clinic for management of inflammatory conditions, autoimmune diseases, And nowadays, they are also routinely used for combating immune-related adverse events in patients who are treated with checkpoint blockade therapy. So the question we asked is, does endogenous glucocorticoid signaling have a role in shaping CD8 T cell effector differentiation in the tumor microenvironment? So the first thing we did is we wanted to look at the protein levels of the glucocorticoid receptors in the CD8 T cells infiltrating the tumors. So the histogram here showing you the staining pattern. The cancer model that we used was NC38 colon carcinoma. And consistent with our previous data, which we've seen in the RNA sequencing, we saw that glucocorticoid receptors progressively increased in the expression from the TIM3 PD1 double negative cells to the PD1 single positive cells, and again was highly expressed in the TIM PD1 double positive cells. We then wanted to see if this expression was different in the antigen specific cells. So because we were using the NC38 OVA model, we could look into the antigen specific cells by staining with an OVA dextramer reagent. So we looked at the expression of glucocorticoid receptor in OVA dextramer positive and OVA dextramer negative cells, and we saw that the OVA dextramer positive cells had higher expression of glucocorticoid receptors compared to the OVA dextramer negative. So this data suggests that glucocorticoid receptor expression could be increased by activation of the TCR in the tumor microenvironment. We next wanted to see if this pattern that we observed in the murine cells would be consistent in the human cells also. So we looked into human colorectal carcinoma, and here too we found that the pattern was consistent. Or in other words, there was a gradient of increasing expression of glucocorticoid receptors from the naive-like cells to the terminally dysfunctional cells having the highest expression. So next question we asked, is the glucocorticoid signaling pathway active in the CD8 T cells in the tumor microenvironment? So to answer this question, we scored the glucocorticoid signature onto single cell RNA sequencing profiles of CD8 T cells from B16 melanoma. And as we can see, there was a gradient of low to high expressing cells. So we wanted to identify what these cells were. So we projected a naive signature and dysfunction signature on the same cell population. And what we found was that the cells that had lower expression of glucocorticoid signature, they had higher expression of the naive signature. And the cells that were very high for the glucocorticoid signature, they were also high for the dysfunctional signature. So this data suggested that the naive cells and the dysfunctional cells are at the two ends of the spectrum, where the naive cells express low amounts of glucocorticoid signature and the dysfunctional cells express high amounts of the glucocorticoid signature. Now, to better understand the relationship between these three signatures, we scored all the cells for the three signatures. And as you can see in this figure, we've scaled the glucocorticoid signature in the x-axis and the naive and the dysfunction signature in the y-axis. So as you can see here, as the cells acquired more of the glucocorticoid signature, they moved out, or they transitioned out from a naive-like phenotype and gradually acquired more of the dysfunctional phenotype. So this data suggests that as cells increase their ability to sense glucocorticoid and there is more of glucocorticoid-induced signaling in those cells, they move out from being naive-like, and there is a reduction in the effector differentiation or effector functions, and they gradually acquire more of the function of a dysfunctional T cell state. So we came up with the hypothesis that perhaps glucocorticoid signaling is dampening the effector T cell differentiation and also promoting T cell dysfunction. So to test this, we carried out a very simple in vitro experiment where we took naive CD8 T cells and subjected them to repeated activation in the presence of anti-CD3 and 28 in the presence or absence of glucocorticoid, the exogenous glucocorticoid dexamethasone. And as you can see here, consistent with previously published literature, addition of glucocorticoid did have a profound decrease in the production of pro-inflammatory cytokines such as IL-2 and TNF, and also interferon gamma. But on the other hand, there was a huge increase in the production of immune suppressive cytokine IL-10. So this data was interesting as it showed that glucocorticoid could indeed reduce the effector capacity of these cells. But we looked at the other aspect of these cells, that is the checkpoint receptors. And what we found was that addition of glucocorticoids did indeed lead to upregulation of checkpoint receptors like TIM3, PD1, and LAG3, However, we found that TIGIT was not upregulated by glucocorticoid signaling. So this data showed that glucocorticoid signaling had an influence on the effector T cell differentiation and was able to generate these cells in vitro, which did share features of dysfunctional T cells in terms of lower production of pro-inflammatory cytokines, higher production of immune suppressive cytokine IL-10, and also expression of checkpoint receptors. So now we ask the question, does glucocorticoid signaling affect the differentiation of CD8 T cells in vivo and specifically in the tumor microenvironment? So to answer that question, we generated mice which specifically deleted the glucocorticoid receptors from CD8 T cells. We implanted MC38 ova colon cancer model into the wild type mice and knockout mice. And as you can see here, indicated by the red here, the knockout mice had lower tumor burden compared to the wild type mice. We wanted to study the effective differentiation and functionality of these T cells in the wild type and knockout mice. And we looked at two different time points. An earlier time point where the difference in tumor sizes between the two groups were not statistically significant, and at an intermediate time point. So the first thing we looked for is the frequency of antigen-specific cells. So as I mentioned before, in our model, we can trace the antigen-specific cells using ovadextromer. And what we found was that there was no significant differences in the frequency of antigen-specific cells at both the time points of analysis. However, when we looked at the cytokine production, we found that the knockout CD8 T cells was able to produce more amounts of the pro-inflammatory cytokine IL-2, TNF-alpha, and interferon gamma at both the time points of analysis. And these are antigen-specific responses as these cells have been activated by ovapeptide. We also looked into the cytotoxic capacity, and here too, at both the time points of analysis, the early and the intermediate, we saw that the knockout CD8 T cells had higher expression of granzyme B and CD107, indicating that they had superior cytotoxic capacity compared to the wild type cells. Next, we looked at the expression of IL-10, and we found that although at early time point, there wasn't any significant differences in the level of IL-10 between the wild type and knockout, at an intermediate time point of analysis, the knockout T cells produce lesser amount of the immune suppressive cytokine IL-10 compared to the wild type cells. And finally, we looked at the expression of checkpoint receptors. So although at an early time point, there wasn't a difference in the expression of TIM3 and PD1 between the wild type and knockout cells, we found that at an intermediate time point of analysis, there was a dramatic reduction in the expression of checkpoint receptors, all TIM3, LAG3 in the knockout mice. Now, one important point that I want to talk about here is that although, if you remember previously, I showed you in my in vitro data, that glucocorticoid signaling was not able to affect or was not able to upregulate the expression of TIGIT in vitro. However, in vivo, you see that the absence of glucocorticoid signaling was able to lead to reduction in expression of TIGIT. So this data suggests that in vivo, of course, glucocorticoid signaling is much more complex than in vitro. And also it alludes to the fact that the glucocorticoid signaling pathway can synergize with several different signaling pathways in the tumor microenvironment and lead to the final output and regulation of the target genes. Now, the final question in this section that we asked is, are these changes that we are seeing in CD8 T cells upon loss of glucocorticoid signaling, are they intrinsic to the CD8 T cells? So to answer that question, we performed this transfer experiment where we took the wild type and knockout CD8 T cells that are congenically marked. So the wild type T cells are marked by CD45.1 and the knockouts have CD45.2. So they were transferred into rag knockout mice along with CD4 T cells. And after the reconstitution of rag knockout mice, 2 days later, we implanted MC38 ova tumors. And then at an intermediate time point of analysis, we looked into the cytokine producing capacity of the T cells, and also we looked at the expression of checkpoint receptors. And as you can see here, although the T cells were derived from the same tumor, so the microenvironment is the same, but even in this condition, only the knockout T cells had superior ability to produce the pro-inflammatory cytokines IL-2, TNF-alpha, and interferon gamma, and also cytotoxic molecules like granzyme B and CD107. However, interestingly, these T cells had lower expression of TIM3 and PD1. So this data now confirmed that glucocorticoid signaling could restrain the anti-tumor CD8 T cell responses by dampening early effector T cell differentiation and by promoting T cell dysfunction in a T cell intrinsic manner. So far I showed you that from our murine data that there is a relationship between the glucocorticoid receptor and the checkpoint receptors and IL-10. So we wanted to see if this pattern would hold true in humans also. So we looked into TCGA database, and what we found is that the mRNA level of the glucocorticoid receptor, so NR3C1 is the gene, did positively correlate with the expression of checkpoint receptors like TIM3, HAVCR2 being the gene for TIM3, and for PD1, PDCD1 is the gene. So for all of these checkpoint receptors, TIM3, PD1, TIGIT, LAG3, and the immune suppressive cytokine IL-10, the expression of, or the level of NR3C1 directly correlated with the expression of these various genes we checked. So with this, we came to the conclusion that maybe glucocorticoid receptor, which is a ligand-activated transcription factor, could directly be binding to the promoter of these genes and transactivating their expression. So to test this hypothesis, we performed a luciferase reporter assay. And what we did is we transfected the 293 T cells with reporters for luciferase reporters for containing constructs for TIM3, PD1, LAG3, TIGIT, or IL-10, and also transfected either the vectors for encoding NR3C1 or control vectors, and treated them with glucocorticoid and assayed for the transactivation ability. And what we found was that glucocorticoid receptor indeed transactivated the expression of all the checkpoint receptors analyzed and also that of IL-10. So this was very interesting, and we came to the next hypothesis that glucocorticoid receptor, perhaps it's not just controlling the expression of checkpoint receptors and IL-10, but perhaps it may be controlling the expression of the entire gene module associated with T cell dysfunction. So to test that hypothesis, we again took in vitro CD8 T cells, we treated them with glucocorticoid, and we subjected them to RNA sequencing. And when we analyzed the RNA sequencing data, what we found was that the genes that were upregulated upon glucocorticoid signaling, they had a significant overlap with the genes that were upregulated in the TIM3 PD1 double positive cells or the dysfunctional cells. And the genes that are downregulated upon glucocorticoid signaling, they significantly overlapped with the genes that are upregulated in the TIM PD1 double negative or the cells which have more of the naive or effector-like phenotype. So with this, I come to the conclusion of the first part. So to summarize, what we saw so far is that there is a gradient of increasing glucocorticoid receptor expression and of glucocorticoid signaling across effector and terminally exhausted CD8 T cell subsets. And what we also saw that the endogenous glucocorticoid signaling has an ability to influence the effector T cell differentiation and promote T cell dysfunction in a tumor microenvironment. Now, the next part of my talk will focus on what is the source of steroid in the tumor microenvironment. Of course, majority of glucocorticoid comes from the adrenal cortex, but there has been some publications showing that the tumor cells themselves also are capable of producing steroids. So we wanted to investigate if there is any local source of steroids in our model also. So this is a very simplistic figure, basically trying to show you that cholesterol is the precursor of all steroid hormones. And the first step in this conversion of cholesterol to steroid hormones is catalyzed by the enzyme called CYP11A1. And CYP11A1 is the first enzyme and also the rate-limiting enzyme. And the presence of CYP11A1 is a reliable marker to say that a particular cell has steroidogenic capacity. So basically, CYP11A1, it catalyzes the conversion of cholesterol to pregnenolone, which is the precursor of all steroid hormones, including glucocorticoids. So the first thing what we did here is to ensure that if there is a production of glucocorticoid or steroids in the tumor microenvironment, we measured the amount of pregnenolone in the tumor tissue and also in the spleen of tumor-bearing mice and tumor-free mice. And as you can see here, we saw that the abundance of pregnenolone was higher in the tumor tissue compared to the spleen. So this showed that either there is an accumulation of pregnenolone or there is active production of pregnenolone in the tumor tissue. Now, if there is local production of pregnenolone or any of these steroids in the local tumor tissue, we wanted to identify the cellular source. So as I mentioned before, just looking for CYP11A1 in these cell populations would be a good and reliable marker for steroidogenic capacity of these cells. So we looked into the expression of CYP11A1 in the tumor cells directly from culture or ex vivo. However, for the cell lines that we use, we couldn't detect the presence of CYP11A1. We looked into the other cell populations, such as the cancer-associated fibroblasts, the various myeloid populations, the various types of DCs, monocyte macrophages, and the T cells. And we saw that all these cells at a different level was able to express CYP11A1. But most interestingly, we saw that the macrophage monocyte lineage cells, they are the highest expression of CYP11A1. So if you pay attention here, this is in log scale. So the production of the expression of CYP11A1 by the monocyte and macrophages was really much higher compared to any of the other cell populations analyzed. So we wanted to study the physiological relevance of this. So what we did is we generated these mice, CYP11A1 flox lysine tree, where the CYP11A1 is deleted specifically from the monocyte macrophage lineage cells. And then we implanted the tumor MC38 ova in these mice, the wild type and the knockout mice. And we saw that the knockout mice had significantly lower tumor burden compared to the wild type mice. So this shows that the steroids, which are derived from the monocytes and macrophages, is a major determinant or a key determinant of anti-tumor immunity. And we wanted to see and look at the functional status of CD8 T cells in these wild type and knockout mice. And what we saw was pretty much similar to what we had observed previously with the glucocorticoid receptor knockout mice. So here too, we found that there was no significant difference in the frequency of antigen-specific cells, but the knockout mice, the CD8 T cells, had higher ability to produce the pro-inflammatory cytokines IL-2, TNF-alpha, interferon gamma, and also had higher ability to express the cytotoxic molecules granzyme B and CD107. Interestingly, it had lower amounts or lower frequency of TCF1, and it had also lower expression of the checkpoint receptors TIM3 and PD1. Next, we wanted to see if, although pregnenolone was produced in the tumor tissue, we wanted to confirm if corticosteroid, that is the glucocorticoid per se, is also produced in the tumor microenvironment. So we measured corticosteroid in the tumor tissue and in the spleen of tumor-bearing and tumor-free mice. And we found that the tumor tissue did actually have higher amounts of corticosteroid compared to the spleen, suggesting that the tumors can locally also produce corticosteroid. And this figure is just showing you the various enzymes involved in production of corticosteroid. So in order to confirm that these monocytes and macrophages, which I earlier showed had higher expression of CYP11A1, if they were indeed able to produce glucocorticoids, we wanted to see if all these enzymatic machinery is actually present in this monocyte macrophages, which were isolated from the tumor. So we looked at the expression of all these enzymes, and we were able to detect these enzymes in varying capacities. Now, to finally confirm that the monocytes from the tumor was able to produce glucocorticoid, we sorted out these monocytes macrophages from the tumor and cultured them in vitro in the presence of an inhibitor called meteropone. So meteropone is an inhibitor that inhibits the action of the enzyme CYP11A1, and then prevents the final step in the production of corticosteroid. So when we cultured these monocytes macrophages in the presence of the inhibitor, we saw that the level of corticosteroid was much reduced. So this shows that the monocytes and macrophages were actively making the corticosteroid. The next experiment we did is we intratumorally administered meteropone into wild type mice. And as you can see here, the mice which were treated with meteropone had lower tumor burden compared to the wild type or the vehicle treated mice. So this shows that inhibiting glucocorticoid production locally in the tumor microenvironment can reduce the tumor burden. So for this part, we conclude that steroid hormones can be produced locally in the tumor microenvironment, and that the monocytes and macrophages are the major producers of glucocorticoids in the tumor tissue, and that inhibition of glucocorticoid biosynthesis in the tumor microenvironment, it resembles the loss of glucocorticoid signaling in CD8 T cells. Now I come to the third part of my talk, where we wanted to study the relevance of glucocorticoid signaling in human diseases. So as I showed you, glucocorticoid signaling, or just the steroid signaling, had an very major impact on anti-tumor immunity in murine colon cancer. So we wanted to see if steroid abundance was also able to impact disease outcome in human gastrointestinal cancers. So we looked at TCGA database, and we found that the patients who had lower abundance of CYP11A1 enzyme had higher survival benefit in both colon adenocarcinoma and also in stomach adenocarcinoma. We next wanted to see the effect of steroid signaling in response to checkpoint blockade therapy. And so we took the wild type and knockout mice, the knockout being the glucocorticoid receptor knockout mice, and we administered anti-PD-1 to both the wild type and knockout mice. And as you can see here, the knockout mice, which were treated with anti-PD-1, had more protection from tumor burden as compared to the wild type. So this shows that the absence of glucocorticoid signaling in CD8 T cells enhances response to checkpoint blockade therapy. Conversely, we did an experiment where we treated the mice with glucocorticoids, and as you can see here, when the mice were treated with glucocorticoid, the effect of checkpoint blockade therapy that we would see in a normal vehicle control treated mice was completely abrogated. So this data shows that glucocorticoid signaling on CD8 T cells, it not only reduces anti-tumor immunity, but also reduces the efficacy of checkpoint blockade therapy. Finally, we wanted to see the relevance of glucocorticoid signaling in human cancer setting. So this is previously published data, and where we looked at the single cells of melanoma patients pre and post treatment with checkpoint blockade therapy. And just to orient you here, the cells in orange indicate CD8 T cells. Here, the orange cells are CD4 cells, and we have the pre and post treatment patient samples, the pre-treatment being yellow and the post-treatment being the pink cells. And we also have the responders and non-responders, the responders in red and the non-responders in blue. So when we projected our dysfunction signature and glucocorticoid signature on this single cell population, we saw that in accordance to our previous murine data, the cells that had highest expression of glucocorticoid signature also had highest expression of dysfunction signature. So our mouse study was consistent with what we see here in humans. But the most interesting part was the patients who had higher or enrichment of glucocorticoid signature, they were non-responsive to checkpoint therapy in both pre and post-treatment samples. So with this, I'll conclude my third part, where we saw that steroid hormone production and glucocorticoid-induced gene programs correlated with poor survival and poor response to immunotherapy. Now, the clinical implications of our study would include the following. Glucocorticoids are widely used to treat immune-related adverse events in immune checkpoint therapy patients, but several studies, initial studies have indicated that glucocorticoid could be used without any negative impact on ICB response. But however, recent studies have shown that patients who are suffering from immune-related adverse event post-checkpoint therapy, if they receive lower dose of glucocorticoid compared to the higher dose, they have a better survival benefit. And also the studies which show that baseline steroid is associated with poor response to checkpoint therapies such as PD-1 and PD-L1. So with this, I will conclude my talk, and this is the summary, where we saw that glucocorticoid signaling, it increases gradually from the TIM PD-1 double negative cells to the TIM negative PD-1 single positive cells, and it's highest in the TIM PD-1 double positive cells, which has dysfunctional phenotype. And we saw that glucocorticoids could be produced locally in the tumor microenvironment by the tumor-associated monocytes and macrophages. And we've seen that if you therapeutically inhibit the production of glucocorticoids from monocytes and macrophages, or if you inhibit the sensing of glucocorticoids and CD8 T cells using knockout mice, as we did, you see better protection from tumor. So this shows that steroids are indeed a major determinant of anti-tumor immunity. So thank you everybody for your attention. And I also want to thank my mentors, Dr. Anna Anderson and Dr. Vijay Kuchru, and all our lab members and collaborators from the Broad Institute and Yale, and also from DFCI, and also our funding sources. Thank you very, very much.
Thank you very much, Nandini. very interesting talk, very complete and thoughtful. So, not sure if we have questions from the chat, but I have my own question that I will start with. So, did you check if the tox transcription factors were affected or were regulated by this signaling?
Yeah, that's a very interesting question. And we are looking into that right now. We have the antibodies now, and we haven't looked so far, but we will have answer for you soon. If glucocorticoid receptor is controlling the expression of tox also. We don't know yet.
Okay, interesting. So here we have another question from Eric. Immunotherapies are often accompanied by colitis, and corticosteroids are indicated to treat colitis. So this will be inhibiting the response to immune checkpoint and decreasing the anti-tumor response.
Right.
Yes. So do you agree with that? Or?
I mean, like I was saying, you know, some studies have shown that it doesn't actually affect the or clinical outcome of immune checkpoint therapy, but several other studies have showed that patients who get lower dose of glucocorticoid, they actually do better than the higher dose. So I think more, you know, studies where you pay closer attention to the dose have to be done in order to come to that conclusion. Although our study would indicate that, yes, if you would administer glucocorticoids, it would reduce or dampen the anti-tumor immunity.
So Mikey FN asks, well, he says, thank you for the talk. Did you also analyze pregnenolone and corticosteroid in the tumor draining lymph nodes?
You mean measure the amount of corticosteroid in tumor draining lymph nodes?
I guess, yes.
No, I haven't looked in the drain. No, I just looked at the spleen and the tumor. And I looked in the blood also, but not the draining lymph nodes.
That would be interesting to see if how is the microenvironment in the tumor draining lymph nodes affected.
Right? Yeah, that's a good question. Yeah, I haven't looked into that.
So Eugenio is asking, does the TCR regulate the NR3C1 expression?
That's a very good question. We've seen that in vitro, upon TCR activation, only TCR activation, the level of NR3C1 goes down. But when TCR is activated along with other signaling that's going on, in fact, even if you add DEX, or we've seen with IL-27, it goes up. So in vitro, if it is only anti-CD3 and 28, the expression of NR3C1 goes down. But in vivo activated cells, because in vivo, the cells receive several different other signals also, there the level of NR3C1 goes up.
Thank you. And Gracie is asking, do the macrophages expressing the glucocorticoid preferentially exhibit a M1 or M2-like phenotype?
Yeah, we're looking into that. And so we think that the monocyte macrophages in the tumor microenvironment, they're producing glucocorticoid themselves, and those glucocorticoid could be acting in both autocrine and paracrine manner. And when they're acting in the autocrine manner, it is pushing them to become more of these M2-like or have more of the MDSC-like type. And those are some things we are working in our lab right now.
All right, and Daniel Heinz, good evening. I was on awesome talk. I wonder why you knocked out the CYP11A1 instead of the CYP11B1.
Why we knocked out CYP11A1 instead of CYP11?
The A1 instead of the B1.
Well, that's a good question. The CYP11B1, it is specific to glucocorticoids, but we knocked out CYP11A1, but that was the most important rate limiting and the first enzyme of the pathway. So that's why we went with that enzyme. But now we are trying to get these mice CYP11B1 also. So then we can have all the mice of the pathway. But yeah, for conclusively to determine glucocorticoid per se, we have to do CYP11B1 also. So we are looking into getting those mice.
All right. Thank you. I think there is no more questions.
All right. Thank you so much.
Thank you very much, Nandini. And we will go on with the next talk. So I give the floor to Eric.
Yes.
Yeah, stop sharing.
Thank you so much. Really nice talk, and appreciate the opportunity to present today Gracie Liu. Chibor in China